So good afternoon, everyone. I am Cheny Yang in 12th grade, and today I'll be presenting my research on the relationship between MMPL4A sequences of clinical and environmental non-tuberculous mycobacteria. So non-tuberculous mycobacteria, abbreviated as NTM, are bacteria that commonly inhabit our surrounding environment. And although most NTM are pathogenic, some of them infect humans and cause pulmonary disease when they're inhaled into our lungs. According to data collected from 1997 to 2007, Hawaii was one of the states with the highest period prevalence of NTM lung disease at 396 cases per million people. And in the map, regions colored in Black indicate higher rates of NTM lung disease, which includes Hawaii as well. M. tuberculosis is a bacterial species that are similar to NTM and cause lung disease to humans, which is called TB, or short for tuberculosis. And as its name implies, NTM are different from M. tuberculosis because they cause pulmonary disease other than TB. Moreover, while M. tuberculosis has been identified as pathogenic bacteria, some NTM species are pathogenic while others are not. And the reason for the differences between pathogenic and non-pathogenic NTM still remains unclear. However, one thing we are aware of is that different genes in NTM may give rise to varying levels of pathogenicity. So M. abscessus, which is an NTM species, has two forms, S, which stands for smooth, and R, which stands for rough. And as you can see in the image, the S forms of NTM have a smoother surface than the R forms. And S forms are also more often associated with non-pathogenic characteristics, whereas R forms display more aggressive and pathogenic traits. The morphology of abscessus depends on the presence of glycopeptidolipid, or GPL, which is located on the surface of NTM cell envelopes and is essential for host to pathogen interactions. When GPL is no longer produced, abscessus converts from an S to R form and loses non-pathogenic traits, such as the ability to form biofilms and sliding motility. Because a gene called MMPL4A in NTM controls GPL production, I hypothesized that this gene could be related to pathogenicity as well. So you might think that NTM is a single species, but there are actually over 200 species of NTM that have been discovered until now. This is why certain NTM species are classified as pathogenic, while others are not. However, as I mentioned before, because the exact source of pathogenicity in NTM hasn't been discovered, not all species have been identified as pathogenic or not. NTM can also be classified as clinical and environmental. So clinical species refers to those that have been identified in patients' lungs or bodies, whereas environmental species are those that have not. Thus, clinical NTM are more often associated with pathogenicity than environmental ones, though this categorization is quite ambiguous because non-pathogenic NTM could also have been discovered in patients' bodies. An example of clinical species is M. abscessus, and an example of environmental species is M. smegmatis, which are both NTM. Therefore, my research question was, what are the differences between MMPL4A amino acid sequences of clinical and environmental NTM species that may be related to pathogenicity? Each NTM has its own unique amino acid sequence for the MMPL4A gene. Therefore, if MMPL4A is related to pathogenicity, there might be a common sequence among clinical NTM that gives rise to pathogenic traits. For my research, I used NCBI, which is an online database for DNA and protein sequences. NCBI contains approximately 108 million DNA and protein sequences, which is a very vast number. Here, I was able to find and download 4 MMPL4A protein sequences of NTM. In addition, I used a variety of bioinformatics programs for my research. Using the 4 downloaded MMPL4A sequences, I used Protein Blast, which is another tool in NCBI that helps us search similar sequences with those we insert. And therefore, I was able to find several other sequences that were assumed to be MMPL4A. After that, I performed a multiple sequence alignment using bioinformatics programs called Genius and Clustal Omega. A multiple sequence alignment vertically aligns gene sequences and indicates how similar they are with black bars, which are regions that have identical sequences. I then converted the alignment results into a phylogenetic tree, which helped me identify how closely the sequences were related. I was looking for homologs among the sequences, which are genes that originated from the same ancestral sequence and therefore are evolutionarily related to each other. Finally, I performed a motif-based sequence analysis using MultiFinder to analyze individual sequences in depth. First of all, because I found only 4 sequences labeled as MMPL4A in NCBI, I had to find more MMPL4A sequences through BLAST. However, I couldn't use all of the sequences that appeared on BLAST because some of them might have not come from MMPL4A, even if they were similar to the original sequences. In order to ensure that the additional sequences I found were from MMPL4A, I established a cutoff for this gene. I did this by creating a pairwise matrix, which shows how similar in percentage each sequence is with another. Thus, I chose the lowest percentage of similarity between these sequences, which was around 75%, as the cutoff for MMPL4A. After establishing the cutoff, I downloaded all sequences that had a 75% or above similarity to the 4 sequences I had originally downloaded and performed a multiple sequence alignment with them. This is the phylogenetic tree I constructed based on the sequence alignments of the downloaded MMPL4A genes. A phylogenetic tree helps us identify evolutionary relationships, and the more the species are related, the closer the lines are connected between them. The clinical NTM are highlighted in red, whereas environmental ones are colored in green. According to my hypothesis, I was expecting to see the clinical NTM species clustered together and separated from environmental NTM, given that MMPL4A is related to pathogenicity. As you can see, some environmental sequences were located in between clinical species, which was different from what I expected. However, the clinical NTM formed large groups together, which was in line with my prediction. I also observed that M. smegmatis was the least relevant environmental species from M. abscessus, which is a pathogenic NTM. Although this tree looks pretty simple, it actually took over a month for me to create it because I had to download all the NTM sequences, identify whether they were clinical or environmental through research, and create the multiple sequence alignments. Because I also wanted to check how different the sequences of clinical and environmental NTM were, I selected the abscessus and smegmatis sequences and additionally performed a motif analysis. The left shows results from smegmatis and the right shows results from abscessus. And a motif is a specific segment of a sequence or a gene that is biologically functional. Each blue bar in the image represents a motif, and its name, position on the gene, and description are listed on the table below. While both abscessus and smegmatis had the MMPL motif, abscessus had 4 additional motifs that weren't present in smegmatis, which are highlighted in purple. According to the sequence alignment and motif analysis results, clinical NTM sequences contained motifs associated with drug resistance. Bacteria benefit from having drug resistance as they help combat antibiotics, which threaten their survival. By doing so, NTM can continue living inside the host body. Since the motifs colored in purple were identified in MMPL40 sequences of clinical NTM, the results supported my hypothesis that MMPL may be related to pathogenicity. As an extension of my summer research, we could further investigate how the protein structure of MMPL4A affects its function. This is a 3D model of the protein for the MMPL4A gene. And since the structure of protein affects its function, we could observe how the changes in MMPL4A protein structure affects the pathogenicity of NTM. Through my K Fellowship research, I was able to learn more about bioinformatics programs and get familiar with downloading sequences, aligning them, and constructing a phylogenetic tree, which are all techniques that I could also use for research later on. For future K followers, I recommend keeping a daily log for your research because it will help you recall what progress you've made throughout your research, which will help you understand your research better as well. I also recommend that you complete your research during your summer before the school begins, because once school starts, it will be difficult to wrap up your research and prepare for your presentation while also managing your schoolwork. And I would also like to thank Dr. Honda and Dr. Strong for directing my research and providing suggestions, as well as Dr. Chan for putting in a lot of time and effort to help complete my research. These are the citations for the images and the papers that I read. And this is, another slide for that. Yeah, and that is it for my presentation. Thank you.